Okay, right. So let me start the presentation. Welcome to today's webinar, everyone. So today, I am Kailing, the Regional Field Application Scientist of Azurebiosystems. And today we will be talking about in-cell Western, which is a very trendy technique right now. How to help or Azurebiosystems help you in terms of generating faster and better in-cell Western data using Sapphire FL imaging system. Okay, right. So this is our company portfolio, and the equipment that we're going to use to help to generate a faster and better result in in-cell Western will be the Sapphire FL biomolecular imager, which is our next gen fluorescence laser-based scanning system. Okay, right. So we are also Azurebiosystems also is a Western blot solution provider. So it covers the basic consumables for your entire Western blot protocol and up to the imaging equipment for different resolutions that you want to output. In conclusion, we have 3 imaging solutions for you. So we have Sapphire FL biomolecular imager, Azure imaging system, and ChemiSolo. So this is application overview of all these 3 imaging system able to achieve, but they of course at different range. The one we're looking at today is Sapphire FL, which is focusing on our high resolution fluorescence scanning, either be on your Blot gel array or plate, which is much more relevant to today's topic, which is in-cell Westerns. And other than that, Sapphire FL can also do in vivo imaging and also photo imaging. For your general laboratory imaging, like for gel documentations and basic fluorescence image, we have Azure imaging systems that covers your basic nucleic acid gels, protein gels, colorimetric signals, chemiluminescence, and fluorescence signals capturing either on your Western blot or gel or other tagged or fluorophorically tagged samples. ChemiSolo will be another very niche product that is dedicated for anyone who wanted only chemiluminescence signal capture. All right, so we have a look at the equipment range that we provide in the Azurebiosystems. Now let's look at in-cell Westerns. Okay, so in-cell Western, we heard about the term for many years. The concept is basically a Western in a plate. So it's not on a blot. And it also can be called as a cell-based ELISA or a plate-based assay. So let us look at the definition. So in-cell Western is a powerful immunofluorescence assay used as a high throughput solution. It's a high throughput solution to quantify target proteins in the cultured cells. In a simplified manner of the entire protocol, what you need to do is to plate or feed the cells in the plate, preferably a glass bottom or plastic bottom that allows you to have a good imaging quality, where you seed the cells in the plate, okay, in the wells, and fix it and permeabilize it in the microwell plate. So this is in-cell Western. We're looking at the protein in the cells, intracellular protein detections. But we have also something called on-cell Western, which is without the permeabilization step. So what you can see is the protein inside or outside of the cells. It can be detected using target-specific antibodies, which is your primary antibody, and labeled secondary antibodies that is compatible with your primary antibody. So you can see it comes in a microplate format. And then how this idea comes out is by combining the specificity of Western blot, okay, and the high throughput feature of ELISA, while allowing you to analyze anything within the cellular context. So what it does is, in cell Western, it can accurately quantify your intracellular proteins, like what we usually do in Western blot. Okay, plus on top of that, you have the repeatability and quick turnaround and the high throughput of ELISA's techniques. So normally we use the conjugates in the near-infrared spectrums, near-infrared NRI spectrums, commonly used to reduce the autofluorescence signals and noises that usually associate with your plastic wares that you use. That is why a glass bottom is highly recommended. Okay. So it combines the best of the two in ELISA and Western blot. Okay. Where ELISA, you can detect small amount of target protein, and it's rapid and easy, and you have exact amount of data that you get from ELISA. And for Western blot, we're normally looking at a wide range of target proteins, like multiple proteins from a complex sample, like your cell lysate, or you need to do some molecular weight analysis or protein modifications of some sort, and looking at the target protein that you specifically look for. The in-cell Western is combination of both of them. Let us look at the clear comparison between in-cell Westerns and Western blot. Okay, so in-cell Western, it fixes up to 96 cell well. Okay, if you're using 96 well plate, 384 wells if you're using 384 well plates. So in these wells, so same thing, you seed the cells, you permeabilize the cells, then you probe with a primary and secondary antibody, and for one or more protein detections. Okay, and the idea here is every well, one well representing one data point. So we're looking at one well as if you're looking at a band on the Western blot. So unlike the Western blot, okay, where you have a membrane, either be PVDF or nitrocellulose membrane, where you load 10 or 15 samples in a gel, then you blot it, transfer it to a blot, and you probe with a primary and secondary antibody, and either detecting one protein using chemiluminescence detection technique, or two or more using fluorescence detection technique. You are looking at them at the arranged manner. So one band will give you one data point. That the data point here could be something like a raw values of the band intensity, something like that. So if you put this into 96 well plate format, it will be every single well, one data point. Okay, so in this sense, the in-cell Western, you do not need to look at the cellular levels. You're looking at the data as a whole in a single well. So when we're going to use in-cell Western and when we're going to use Western blot, there will be a decision making. Either you want to do a high throughput analysis of a well-characterized protein. Okay, for example, you are doing drug screening, molecular screening, or pharmacology studies. One example that I normally use will be, let's say you have cancer cell lines, okay, can be human cancer cell lines, and you want to test a series of anti-cancer properties, okay, and then by increasing dosage, and not only the increasing dosage, but different type of compound as well, because you need to do it in a large quantity. By screening one or two or more cell lines, cancer cell lines, you need to be very high throughput, quick, and consistent. This is where the in-cell Western comes in, because you will know which biomarker they're looking for, which will be the cancer marker. And by analyzing the target protein or the marker they're looking for, like the cancer marker, and then you normalize with your, let's say, housekeeping protein or cell stain, that's normalization control, then you will get a set of data that is in a single assay that you can compare dosage to dosage or increasing dosage or different type of compound as well. So this is what in-cell Western allows you to do. Okay, so it's high throughput analysis of characterized protein. Can be applicable for drug screening, okay, active compound screening, pharmacology, or any kind of cosmetic product screening that is allow you to quick and at a high volume. And Western blot, on the other hand, which is a very generic technique we use in most of the lab, it is very good for discovering new, novel, or characterized protein that is not well known. So we can identify a new protein or identify or characterize a protein or molecular weight analysis of that target protein itself. So this is more like a discovery and characterization analysis on that you will use a Western blot or something that is not required high throughput analysis. Okay, so it's commonly used in academic research or discovery of any novels or modification on the proteins. So that will be a Western blot site. Okay, but if it is going to high throughput, that will be in-cell Western. Okay. Next, let us look at the table format. What in-cell Western allows you to answer the question for. So, of course, traditional Western blotting allows you to do protein discovery, okay? Protein discovery definitely, and molecular weight analysis because of the protein ladder. We know how the protein sizes and possibility of having any protein modifications, okay, or glycylation or something. And both in-cell Westerns and traditional Westerns, okay, allows you to profile the proteins, means you can access the expressions, okay, and activations, okay, means whether the protein is expressed or not under certain condition, having higher or lower intensity or expression based on different condition as well. And it also both allows you to do multiplex analysis. Here, in traditional Western, we'll be using the fluorescence detection technique because you can detect multiple proteins on the same blot. And on in-cell Western, you will have it will be done in the plate. So both that allows you to do simultaneous access and quantification analysis on multiple proteins within the same sample. So for in-cell Western will be within the same well, and traditional Western will be across the entire blot, also within a same sample in the lane. Okay, and of course, both allows you to do quantitative analysis of the protein levels and modifications, which allows you to understand the dynamics of the protein within the cells. Okay, but what traditional Western cannot provide, but in-cell Western is very good in telling, will be in situ analysis, where you do not need to disrupt the environment of the cell culture environment. So because in normal Western blot circumstances, we need to harvest the cells, by trypsinizations, and then we need to lyse the cells and then spin down and get the cell lysate. And there are certain studies or assessment that require the cells to be in and undisrupted as possible, because any process in between from the moment that you harvest the cell to the end of getting the cell lysate may change the expression of certain target proteins. For example, phosphorylations or so on. As much as we want to control it, there will certainly be any discrepancies in terms of that. So in-cell Western, what it does is, because we do not change the condition of the cell living conditions, we proceed to the fixing and permeabilization straight away. So that will allow you to profile the proteins within the cellular environment. And of course, finally, it gives you a high throughput data where you can perform a large scale study and also more efficiently in terms of drug screening, compound screening, and so on and so forth. So with in-cell Westerns, you can answer questions about your protein levels inside or on the surface of the cultured cell, and how the protein's level changes in response to your cell treatment, either be a drug treatment or that either be an inducing condition or worsening conditions. Okay, and because it is high throughput and is streamlined nicely in an array format, assay format, it is also a quantitative assay. It can be used to answer questions that normally require multiple replicates or testing different conditions. Let's say you have different groups and harvesting time point, that kind of situations. In overall, it gives you a big result or lot results in an efficient and very cost-effective manners. Okay, so some of the examples is, as I mentioned before, will be like drug screening and so on and so forth. One of them will be like, let's say, cell signaling studies. Okay, so for example, protein phosphorylation levels, like kinase signaling, like MAP kinase cascades. So this is like a mass screening about a certain thing, sort of screening them, and also a large quantity of amount. Or they can study the abundance of the cell surface protein, ligand binding, surface receptors, internalization. Those are more for cell signaling. And it can be also applicable to virology. Okay, virology studies like access the viral titles, like how much replicates of your titles responds to the drug that you induce on the wells. Okay, so it is a very good high antiviral agent screening for that. Okay, and of course, drug screening for your markers like progressing diseases or certain kind of using silencing RNA or therapeutics as a compound to do that. So this is in-cell Westerns. Like our Western blot and in-cell Westerns and any other techniques that we use in the laboratory, we do need to have normalization controls, because in real world, okay, so the amount of the numbers of cells, as much as we try to control within the wells, okay, it will definitely have differences in the numbers of cells, because this is human, we are not robot, so we can't really control. This is something that we cannot be negate. So you will see the errors or the expression changes depending on the number of cells. Sometimes we cannot really attribute the changes in protein signals. Is it due to the number of cells, or is it due to the experimental conditions? That is why, like our normal Western blotting, you will need normalization control in the in-cell Westerns. Okay, so normalization is the use of internal control over here. Okay, so normalized volumes. Then that is mathematically account for the differences of the number of cells within the well. So in in-cell Western, the only thing we can control will be the number of cells. So we assume that the number of cells, as we normalize the number of cells, wherever changes or expression changes within the well itself will be the result from the treatment itself. That is what we assume. Okay, so that's why the normalization will normally using total cell or cell stains in in-cell Westerns, as compared to traditional Westerns, where we normalize against, let's say, a housekeeping proteins or a total protein signal in a lane. Okay, in in-cell Western, we are normalized against a total cell signal within a well, within each individual wells. So we call it the well number X. So I will not go into details about the calculation and so on, but basically it's the same normalization technique, like how we do in Western blot. Means you normalize against the housekeeping or the total cell stains, and that is your normalized value of your target proteins. Then you do your group-to-group comparison and statistical analysis. So just like in-cell Western, there are two options for you to do your normalizations. You can use your reference protein, okay, and you can use a cell stain. So reference protein is something that if you, let's say, cascade your research or your techniques from normal Western blotting to an in-cell Western, if you want to reserve the reference protein that you already validated the expression changes is a constant throughout the entire experimental conditions and treatments and so on and so forth, then reference protein will be a good normalization control for you. But if you move on to already using interwesterns and then you want to have a standardized normalization control, there will be normally your cell staining, either be a DNA stain, either be intracellular cytoplasmic staining, or total cell staining. So you are staining the entire cells that is serving your normalization control. So the good thing here is it actually removes the changes, normalize the changes of the cell conditions, either they are dead because of the treatment or so on. Okay, it normalizes against that. So it's rather more consistent, but it has a very shorter dynamic range. Okay, so we have talked about interwesterns, all right? Then let us move on and look at how Sapphire FL are able to provide you or give you the faster and better interwestern data. So interwestern data, we know that it's going to perform in a 96 well plate. Let's say it's a 96 well plate. Okay, so Sapphire FL we have here is focusing on fluorescence scanning and flexibility. So it has is highly sensitive because of the detection, the technology. We have customizable and user changeable optical modules, which I explained in the later slide. Then it can be used in a wide range of samples because of the adjustable Z-plane and also different resolutions that you're going for. And then it also allows you to have options to do, not the options, but you can do live animal imaging, okay, because it has a gaseous anesthesia port for you. Okay, so firstly, let's look at how the system, the equipment allows you to detect a very good fluorescence signals. So in Sapphire FL, it uses both PMT and APT that has the best quantum efficiency at its own, I'll say, visible and also near infrared range, okay, that is give you a very good back signal to noise ratio, and the detection is very good, okay, compared to CDD and the scientific CMOS. It is a point-to-point scanning, okay, so it's a laser diode, gives you the excitation, light to your fluorophore, and then the fluorophore emits the light detected by PMT or APT. So it's a pixel by pixel scanning, okay, to your fluorescence sample. Okay, unlike the other competitor, either be using a line scanner or CMOS detectors. So the combination of laser diode and PMT and APT are very good in this sense. It gives you the higher sensitivity that we know on the market in terms of the laser scanning system range. So the flexibility here will be something that is more suitable for interwestern users, okay, which is the flexibility in dye and fluorescence signals that you can image. It has 3 slots. Sapphire FL has 3 slots, allow you to do 3 channels imaging at the same time, and it supports a wide range of wavelengths, covering from a UV range 375 and excitations and up-to emission at 900 nanometer wavelengths. So you can see the lasers and emission filters pairing here that we listed that is all ready to for you to select. So we have the laser at 375, okay, in the normal 488 channels, okay, 532, like Psi 2, Psi 3, Psi 5, and then near infrared channels. So the one notable thing about fluorescence imaging instruments is they usually comes with fixed channels, like let's say 488, 532, Psi 2, Psi 3, Psi 5, and near infrared. But because these fixed channels, they kind of limit the development and adoptions of new fluorophores, because as we progress as a new techniques, you may using a different plasmid or different cell type that is already fluorescently tagged that you want to image it. Okay, so what Sapphire has is it doesn't confirm with such standard pairing. So it is a customizable pairing. So you can see here we have the filter allow you to plug in and plug out. Okay, so you can mix and match the laser and the emission filters. With that, Sapphire FL able to image DAPI. DAPI is a very good and cheap staining on the cell. So if you have samples that you have DAPI signals, a Sapphire FL able to image that. DAPI, as we know, it requires UV excitation. So this is just example data where we have a DAPI stain in Azurebiosystems using DAPI. Okay, which if you use a normal 488 standard channel to image, you cannot get the result. But we have a custom optical module for the DAPI, which will reliably and sensitively image the DAPI stains. So this modular design is an uncoupling design that you can uncouple the excitation from the emissions. So what you can do, you can mix and match, have more than one combination. One example you can see here is the standard 488, which will have the laser 488 emission filter in 5 to 13. But in yellow fluorescence protein, the emission filter is ranged from the 534. So we have a custom emission filters. That means if, let's say, in the future you already have these 488 modules, but one day you happen to have a strain or a plasmid or a transfection kit or some sort that is using yellow fluorescence protein or a cell line that has yellow fluorescence protein, what you need to do is only purchase the emission filter and swap in, and then you can image yellow fluorescence protein. So that's the whole modular design for this for the maximum flexibility. Another thing will be mega stroke dyes. So normally in fluorescence signals, either be Azurebiosystems, fluorescence, or ICC or IHC. So ideally, we want to have the excitation and emission wavelength to sufficiently separated, okay, from the excitation light. So we don't want to bleed between these two wavelengths. So where you have like later it will translate into a high background and so on and so forth. So the distance between the excitation and emission maxima is called as Stokes shift. So you can see in table 1, the normal Stokes shift that we see in sheet C, side 2, side 3, side 5 are ranging between 10 to 40 nanometer wavelengths. So this is standard what we have in fluorescent microscopes, okay, in the normal fluorescent laser scanning system and so on and so forth. But there are more development in digging, designing, and looking for dyes that have larger Stokes shift like this. So you can see here there are 2 dyes, okay, we listed here. The Stokes shift is around 130 and 162 nanometer wavelength. Okay, so the Stokes shift that is greater than 80 nanometer wavelength, it will minimize the crosstalk between the excitation light and the detection of the emitted fluorescence during the imaging, where you have a better signal to noise ratio and imaging quality. So this can be done by pairing the excitation and emission filters, the modular design that we have in Sapphire FL. So let's look at the results in this Blot dyes, mega Blot dye. We have it on the Western blot. So you can see by the 488 excitation plus emission around 624, so we can image the dye reliably, okay, without much crosstalk. Next thing will be on the application and sample type flexibility. This will be much more on the adjustable Z-plane or focus that's up to 6 mm and also up to 5 micron resolutions. So let's look at the first thing, the adjustable Z-plane focus up to 6 mm. So what it means is Sapphire FL allows you to image anything from 0, which is your blot, 0 mm, focus, up to wells, okay, well plate is around 3 mm focus, or any other custom plate or different plate that you can custom the focus for. So All this is adjustable. The Z-plane focus, you can adjust it, okay? And this adjustable focus allows you to image samples with different depth. So the depth can be applicable to different samples or penetration into the samples like your mice or certain, let's say, zebrafish and so on and so forth. And then we have also the Sapphire FL allows you to merge, okay, the images or transform the data into a G file. So you can see, look at the expression changes across different focal plane. So it supports in vitro to in vivo studies like your membrane, fixed cells in the well plate, okay, tissue section select, or ICC, whole organ, or even live animals. So first thing, 4-plex fluorescence Western blot. So this is a 4-plex fluorescently labeled Western blot that target 3 target proteins, okay, plus assuming 1 housekeeping proteins. So it is very good for quantitative. You are looking at 3 set of data that is plus a normalization control within a single blot. Okay, so that is for your fluorescent Western blot. And Sapphire FL can image fluorescently labeled gel imaging, okay, that can be a fluorescently labeled samples like your plasmid and so on, or extraction for a certain samples, or can be you label the gel contains, or can be a nucleic acid or protein. You also can image Comessi gel, Comessi stain gels, okay, by using the 685 modules, okay, right? So which is the BSA that is separated by SBS page. Sapphire FL also allows you to quantify live and dead cells, okay? So we are looking at the overall fluorescence signals of these 2 channels, then we quantify and normalize it against each other, then we get the percentage of viable cells. And you can also use it to check the transfection efficiency or checking or trying to see which transfection kit that is best for your sample. So this is the 24 well plate scanning that we done testing 3 different transfection kits that we're looking at. We are fluorescence protein here, and with a negative control here, we can see by our naked eyes, very, very clearly that this transfection kit is the best for our cell lines. And for liquid culture of cell expression red fluorescence protein, this is on the 6-well plate, tissue array that is multiple tumor matched tumor and also normal tissue microarrays. Okay, we're looking at the 488, which is the F-actin probe that is indicating the tumors. Microarray and also in-cell Western. So by performing the in-cell Western, so you can see this is a 3-plex in-cell Westerns. Okay, the plate focus setting is 3 mm. We have 1 to 2 serial dilution of the stellar cell line. So please bear in mind this is an example to showcase the in-cell Western data that you can capture on the Sapphire FL. So performing the in-cell Western on 96-well plate, so you can accurately measure, okay, the intracellular protein expression in situ, is within in the cells without changing much of the environment. So this is a very high throughput methodology. So you can access multiple stimulation, endpoint, protein of interest, or even time point setting, depending on your experimental designs. And you can have replicates. Unlike the replicates we normally have on Western blot, maybe you can do 1 or 2. This is like more. You can have more replicates for every single group. Okay, so by means, these are not only give you high throughput data, it actually increase the number of data points that you generate by every single assay, therefore lower the overall standard errors of mean and also error bars for the assay as well. And then, so we using here, this is near infrared channels, 700 and 800 channels. And then we have it normalized against the total cell stains. Okay, so it offers these high throughput data. We also have sample holder for plate holder and slide holder. So what it has in plate holder in Sapphire FL is we have a preset protocol where you select the plate holder. The scan region will automatically display on the scan in software, and then you can see here, okay, on the bottom right, every single 96 well plate or microplate, okay. So scanning at 100 micron resolutions will only take around 7 minutes. So it is the simultaneous scanning, means all 3 channels at the same time will take 7 minutes for every single in-cell Western plate. So imagine that you run your in-cell Westerns overnight, in let's say a primary antibody overnight, then you do your secondary antibody scan incubation, and by the afternoon you will able to get the set of results by the end of the day, by day 2. And then each of the plate will only take 7 minutes to scan. Then you have your 3-plex data, which is maybe 2 of your target protein plus 1 normalization control. Okay, so this is how Sapphire FL gives you sensitivity and speed in in-cell Western imaging. Okay, so let us look at the steps where we do the in-cell Westerns. Okay, so it is like the ELISA, okay, but not the antibody in the at the bottom of the wells. Right now, is you seeding the cells at the bottom of the wells. Okay, then you fix the cells. Okay, you fix the cells, permeabilize in the culture dish where they were grown, and then you detect the blocking primary antibody, secondary antibody, then you do your total cell stains. So this is much like our traditional Western blot, but it is in a plate. Therefore, we do need the permeabilization solution. But if you, let's say, your protein is not intracellular protein, it's on the cell surface, then you do not need to do the permeabilization step. That will be detecting on the cell surface then. Okay, right. So the steps is much like the normal Western blot. Okay, blocking primary, secondary. Okay, then you wash. But between the steps, okay, normally the in-cell Western will be labeled with the near-infrared fluorophores because they are less subjected to interference for autofluorescence and very good noises. And then normally a nuclear stains or like total cell stain will be used for normalization control. So each of these steps, like any other techniques that we use in the laboratory or R&D, the reagents and steps must be optimized and validated. Here we have Azul. We do offer a full set of kit, okay, that contains all the reagent necessary to stain the whole cells, okay, for total cell normalization plus simultaneous detection of 2 biomarkers. Okay, so you enough for 596 well in-cell Westerns assay. So this is a kit with 2 biomarkers in near-infrared channels, okay, or optionals. There will be different models, and 1 total cell stain for your normalization control. So this is 3-plex in-cell Western. This kit can use to do 5 in-cell Western assay, means 5 times 96 wells. So this is a kit, how it looks like. These are the solutions that allows you to, in the kit, where you have your permeabilization solutions, you have your blocking solutions, okay, and also the total cell stains, which is linear, and also good for the normalization control, and the secondary antibodies, okay, right. Here's a step, okay, in how to do the entire in-cell Western. So it's a kit. So anyone who is interested to start up their in-cell Western, okay, this is a full set of kit that is without the primary antibody and of course without the samples, okay. So it provides anything starting from your fixing onwards, means your permeabilization, blocking, okay, then so on and so forth. So let's look at the steps then. The first thing you do need to seed your cells, grow it, okay? At least you need to incubate overnight, okay? Allow the cell to seed again, and then seed and adhere to the bottom. Then you fix the cells, permeabilize, and then also you block, okay, do the blocking steps. Permeabilization is necessary if you are detecting intracellular protein, but if you are detecting surface protein, you can omit the permeabilizations. So think of entire step like your ICC in immunocytochemistry. Yes, so ICC. So it's the same concept, but every single well is one data point. One well, one data point. We're not looking at microscopic cellular levels. Okay, right. So after fixing permeabilization blocking, that is for your intracellular protein detection, incubate with your primary antibody and also total cell stain solutions. Wash, then incubate with the near infrared secondary antibodies, okay, 700, 800 channels. Then wash away. Then you can scan the plate, okay, at this is I think the side 3 channels, and then the near infrared channels for 7 minutes at 100 micron resolutions. So every plate is only taking 7 minutes. So in this case, you can imagine how it expands in a high throughput manners, because you don't need to only do one plate. You can do up to 4, 5, or even 10 plates per day, and then you only need to scan 7 minutes for every single plate. 100 micron is more than enough for you to get image like this. Okay, so this is an example of the image that we get from that we're using the kit. Okay, which this is also available in the format of application notes. If you're interested, please feel free to go to our website to download it. So we have, it has the sensitivity of the total cell stains, so you can normalize at the lower concentration as possible, covering different number of cells. Then enhanced accuracy, because we have the best near infrared channels that is very good in eliminating autofluorescent signals and crosstalk as well, and versatility in combining different conditions. Okay, right. So this is a merged image of your in-cell Westerns. So near infrared plus 1 normalization channels. So this is the linearity, signal linearity of the total cell states we have in the kit, covering from 0 to 30,000. Oh, sorry, this is 25,000 cells. Okay, they're having a very good linearity. All right, so this is applicable to HeLa cell lines and certain cell lines that we tested. But for your cell line, you may need to test out the total cell stain linearity, so you can have a rough idea that is linear. Then you move on with your in-cell Westerns. Okay, right. So our Azurebiosystems in-cell Western kit is optimized and also validated to work together with Sapphire FL. So it is an 2 near infrared 700, 800 target plus 1 normalization control, which is a total cell stains. Kit is a total package of the in-cell Western kit, covering your permeabilization, blocking, and washing, also secondary antibody. Okay, for 596 per plate. So here's also the example that we shows. Before, this is the data for the tubulin. Okay, A, which is tubulin at 700 channels. Okay, this is B, GAPDH detected in 800 channels. This is our Azurebiosystems total cell stains at, which is, this is red, this is green. Yeah, so this is blue, yes. So at 532 channels, and this is a merge image. So you can see that the expression changes at different column. Okay, so if they are specific, they are very clean. Okay, so you minimize the crosstalk when pairing with the Azure Sapphire FL imaging together with the in-cell Western kit. So this is more about someone who wanted to try, and then we have already a commercially available kit for you. Sapphire FL also allows you to image glass slides, tissue section slides, up to 5 micron resolutions, and where we have a scale bar allows you to blend in on the image, so you can put it in your manuscript for publications. So what it provides in a single field of view, like what you did on the confocals or fluorescent microscope, but This is without stitching. So you scan the slide in one go, you get a single image. So it's a very good single field of view image for your presentations or manuscript representative images. Imaging of living small animals. Here we have examples of mice, okay, because we have around 4 centimeters of clearance. So anything thinner than 4 centimeters, so you can fit in. So either the mice, zebrafish, mosquito, either kind or any kind of sample and living samples. So we have 5 gaseous anesthesia pods for you to anesthetize the small animals and then let them sleep throughout the entire scanning. So this is what we done on imaging the living animals, small animals. So it's what we're trying to show here is without the fur, once we save for the fur, what the Sapphire FL are able to give you is a very clean background without autofur signals. So this is what we want to prove. And these living small animals or organisms imaging, so you can use it to do any type of scanning for your cell migration studies, okay, preclinical drug testing, or tracking over time for either tumor growth or tracking the migration of certain marker or certain things throughout the entire body of the samples. So this is an example of in vivo tumor imaging that we've done. Okay, so the tumor is actually on the side that is facing the glass slide, the glass scan bed. So we have 41 cells expressing the red fluorescence protein. So the image in 100 micron resolution after 11 days. So these are tumor progressions. We have it after 4 days post-injections, 11 days post-injections, and 14 days post-injection. This is harvested. So we try to tally the growth within compared with the one that we scan at time point manners. And here's a GIF file showing the confocal imaging of these tumor growth of all are injected with 41 cells, but there are 2 of them, okay, 2 or 3 of them without any they have RP, and then those that control is they're having the tumor growth but without the red fluorescent protein. So you can see there are the signals there, they are growth but it's clean. So this is what we're trying to show in Sapphire FL. It's not only showing the data, but it is giving you a very good sensitivity and clean, a very high signal to noise ratio data. Ex vivo dissected tumor imaging. So this is what we're trying to show, okay, and in grayscale as well. Okay, so all the fluorescence signal imaging in Sapphire FL, okay, is supporting extended dynamic range. Okay, so here I want to show what we have in a complex data. Let's say you have overexpressed samples, a wild type, and knockout or knockdown samples, where you have to quantify them on a single assay. Here will be on a Blot. Okay, so in normal circumstances, if you generate a normal standard 16-bit raw TIFF file, the dynamic range may not able to cover the high intensity data or signal at the same time. So by extending the dynamic range, where Sapphire FL allows you to select and then capture the signal in extended dynamic range, you will have a single image that is covering all the signals without saturation. So without saturation means you can analyze the data already. So you don't need to abandon the data. So this is EDR, extended dynamic range. So it doesn't only apply to Blot, it can apply to any other array or assay that you do, even in cell Western, because you may not know the expression, some are high or some are low. So this is something that is optional for you to choose in the software. Okay, right, so this example, without the EDR, okay, extended dynamic range, some of the band will be saturated. But when we extend the dynamic range and capture the fluorescence signals on a Western blot, now we have an image that is non-saturated. So that means you can analyze the data. So one of the declaration is, of course, this data will be not 16-bit, will be either 18, 20, 22, or 24-bit image, file image. So you do require our own Azure Pro analysis software to analyze the ADR image. Sapphire FL also allows user to detect radioactive samples. For example, you have radioactive isotope label for, let's say, hydrogen-3, carbon-14, phosphorus 32, or sulfur-35. Those are targeting or metabolite kind of testing, or try to check the trafficking of a certain like protein, lipids, or nucleic acids. Then Sapphire FL able to detect that. So it uses the PNT, the best detector for the radioactive samples, a very targeted one for your radioactive label samples. So this one, the data we showcase, then will show the detection of the radioactive sample, the linearity. So of course, if we know the Sapphire FL is specifically for fluorescence laser scanning system, but if you need a chemiluminescence imaging module that is complement with your fluorescence scanning, we do have chemiluminescence module. So this chemiluminescence module is only able to capture chemiluminescence signals with color marker. And it also has densitometry package, allows you to generate a standard curve based on your data with the standard plate that the kit provide. Then you can quantify the spots on bands of your parametric protein gels like chromacy or BSA or silver stain or any other extra filmed image for Western blot or Northern blot or Southern blot. So there is densitometry analysis. So here we go, we have Sapphire FLs. So it's flexible in detecting fluorescent signals in a standard, the RGB near infrared channels, and also other kind of optionals, which is like yellow fluorescence protein, red fluorescence protein, M cherry, and DAPI, and so on and so forth. And it allows you to detect signals that different focus up to 6 mm. So you can capture the signals in different depth. Let's say Western blot, plate, okay, and gels, and also animals. So the penetration up to 6 mm, and then you can have up to 5 micron resolution. So you can image anything that is at high resolution for, let's say, a microarray or your tissue section slides. So here are some application notes that we have progressively released that is under Sapphire FL applications. So we have recently IHC image of tumor cell array on Sapphire FL. So this is array imaging. So it's scanned at 10 micron resolution. And this is immunohistochemistry slide multiplex by scanning at 5 micron resolution. In vivo imaging and monitoring of red fluorescent proteins plus red fluorescent protein tag tumor cells in mice, okay, using the Sapphire FL. Okay, multicolor fluorescent imaging of culture cells, which is your AO and PI assays, okay, and imaging yellow fluorescent protein expressing cells, okay, and that is for the transfection checking. And also the mega stope dye that is have dyes with large stope shift like this, that is very good in terms of background signals and increased sensitivity. This will be only uncoupled laser scanning system can do. One of them will be Sapphire FL. And of course, one of our laboratory with the academia, there's on their ongoing Zika virus study, there's one of the bimolecular fluorescence complementation by FC assay in determination of in vitro protein ligand interactions. And these are all the application notes are available on our website. We also have publication booklets, okay, that covering all these applications, including in-cell Westerns as well. In fact, we have a lot of technical notes on explaining how to do in-cell Westerns and the choices that you made when to go for Intel Western, like between ELISA and Western blotting, when to go for Intel Western and how to do, and maybe something you need to be considered or tweak around if you're moving on from that. So we have tons of information on our website. Please feel free to check it out. And here's the Sapphire FL applications. I'll just spare all the lists. Basically, anything within 6 mm depth, you'll be able to image it. Okay. Then this is summary of the Sapphire FL. We highly recommend user or anyone who do Intel Western to try it out with Sapphire FL because it has very good, it gives you sensitivity and speed as well. So 7 minutes for 196 row plate scanning for 3 channel fluorescent signals, which is very good. And the signal to noise ratio is very, very good as well. So we have installations, which hold on, I think for the past 6 months, okay, not past 6 months, past 3 or 4 months. So these are the installation all over the world right now. And yeah, so this is what we have in the Azurebiosystems, where Sapphire FL is our advanced and high resolution fluorescent scanner that we dedicated for Intel Western users. And thank you for your attention. Okay, I am Kylie. If you have any questions, please either you can speak in Korean to our MC Dongjun so we can do translation.
Yes, thank you, Karin.